Welcome to the Stem Cell Spinal Cord Repair Design Project. In this project, we designed a production scheme capable of producing neuronal cells from human mesenchymal stem cells to treat patients suffering from a spinal cord injury. There are about 285,000 people in the United States living with a spinal cord injury, with 17,000 new cases added each year. Injuries from spinal cord trauma can range from some loss of motor function to total tetraplegia. Patients with minor loss of motor function pay around $220,000 in hospital bills during their first year living with the injury and about $27,000 every year after their first. On the other hand, patients experiencing total tetraplegia on average pay about $740,000 during their first year and up to $133,000 annually after that. Additionally, the treatments given to patients with spinal cord injuries only treat the symptoms of the spinal cord damage rather than treating the injury itself. Stem cells have the potential to change how spinal cord injuries are treated by treating the injury itself instead of the symptoms. It is expected that patients with spinal cord injuries whom are treated with stem cell-derived neurons will pay less both in their first year and for the rest of their lives. The stem cell type we propose will work best for spinal cord injuries and in the scale-up process is the human mesenchymal stem cell. These cells are collected from the fat tissue and bone marrow of allogeneic donors, which are then frozen and stored until they are needed. Human mesenchymal stem cells, or hMSCs, can be differentiated into neuronal cells found in the central nervous system by introducing differentiation factors into the process, which we will discuss later. Additionally, stem cells have shown success when grown in a scaled-up process rather than on a lab benchtop. The first step we took in our design synthesis was to research the current and projected behavior of the stem cell production industry, as well as any potential competitors in the United States or across the world. It was predicted that the global market for stem cells would have about a 25.5% annual growth rate from 2018 to 2022. In the chart at right, it breaks down how each category within the stem cell industry will grow. Stem cell production to treat cancer was projected to increase the greatest, while the stem cells for tissue engineering and immunochemistry, the category that our process will belong in, will increase the second most. According to the chart, stem cells are gaining traction across the world, which will make finding an investor easy. There are several stem cell-based products across the world, but none of them are used to treat spinal cord injuries. In the United States, Osiris Therapeutics is the only company that sells medical-grade stem cells, but their products are only useful for treating burns, minor open wounds, and cartilage industries. Across the world, companies like Adipose in the United Kingdom, Pharmaceutical in South Korea, and Sonata in Australia have their own stem cell treatments for cartilage injuries, acute myocardial infarctions, and kidney disease.
Various options were considered for our final process. Different methods and techniques used in industry were compared to one another for both the upstream and downstream portions. First, there were different types of bioreactors that we could have used, such as a stirred tank, wave, or vertical wheel bioreactor. Media considerations were also important to the final design, such as using a media with or without animal components. hMSCs are anchorage dependent, meaning that the cells need a surface to grow on. Therefore, different options included using adherent or pseudo-suspension methods. Various design constraints included maintaining the correct temperature, pH, agitation rates, shear stresses present in the bioreactor, and the amount of dissolved oxygen in the system. Our final process was based on what would maintain these constraints and what would provide the highest titers. Since hMSCs are mammalian cells, an optimal pH of 7.2 and a temperature of 37 degrees Celsius will be used. hMSCs have traditionally been grown in tea flasks and multi-layered culture vessels, but it was found that using this process was not ideal for larger productions of the hMSCs. Therefore, microcarriers will be implemented to create pseudo-suspension growth conditions. In addition, hMSCs are known to be sensitive to hydrodynamic shear stresses, which causes cell lysis, decreased production of product, and early differentiation in the hMSCs. To decrease the shear stresses in the system, a vertical wheel bioreactor will be used. The media that will be implemented is a serum-free, chemically defined media to minimize the risk of contamination with pathogens such as mycoplasma bacteria, prions, and varying zoonotic agents. Lastly, since the hMSCs are grown on the microcarriers, it was found that the best methods to harvest the cells included using chelating agents and a filtration system. Next, we built the model for the process using SuperPro Designer. There are 4 major steps that were fulfilled in this process. The first step is preparation of the hMSCs, where the initial set of cells were received from a donor, thawed, and washed to remove the cryopreservation solution. This step involves process units P1 to P3. The second step consists of increasing the cell density. The units used in this procedure consists of shake flasks, a wave bioreactor, and 2 production bioreactors, sizes 70 liters and 700 liters. These are shown as P4 through P7. Microcarriers were then added to the bioreactors to promote cell growth in suspension rather than adherent cell growth. Gas compression and air filtration were done to oxygenate the bioreactors to promote the cell growth. The third major step of this process is downstream purification. These steps are shown with units P8 through P10. In the downstream purification, 2 major filtration steps were done to concentrate the hMSCs. The first is dead-end filtration to remove the microcarriers, and the second is diafiltration to remove enzymes and other waste. The fourth major step in this process is finishing, which is shown with unit procedures P11 through P14. In the finishing steps, neuronal media was added to facilitate cell differentiation. The cryopreservation is then added, and the cells are then packaged into vials and are frozen using liquid nitrogen. All of these unit procedures are necessary to produce a purified product that can be easily stored and be in compliance with FDA guidelines. Several assumptions were made while making this process. One important assumption was using a stoichiometric conversion approach for the growth of the hMSCs. The equation used was equal to 145 glucose plus 102 oxygen, which converts to 130 carbon dioxide plus 12 cells plus 105 water molecules. This was based on the equation for cellular respiration, since hMSCs are mammalian cells. We assumed that the growth of the hMSCs were directly related to this equation. Another assumption was using large general bioreactors in the model because SuperPro did not have an option to use vertical wheel bioreactors, which were in the original plan. Process time for each operational procedure was set to 60 minutes, which may or may not be accurate in industry. This was based on an educated guess. Lastly, flow rates for each procedure were based on literature, with slight changes in what we thought would be appropriate for our process. For each model block, settings in SuperPro Designer were kept and not altered. Operational data was slightly changed. For example, most of our equipment was disposable, therefore steam in place and clean in place settings were omitted from those sections.
A techno-economic analysis was used to determine the facility startup cost. The analysis was generated with help of the SuperPro software. Direct and indirect plant costs were considered. One of the largest contributions to cost was equipment, which accounts for $4.3 billion. This price is generated considering reusable bioreactors. However, we believe that disposable bioreactors should be utilized for this process. Disposable bioreactors will increase the safety of the product and the efficiency of production. Disposable bioreactors will decrease the initial startup cost while increasing the operating cost significantly. The simplifying assumption, a multi-use equipment was made because disposable bioreactors were not able to be estimated accurately. Overall equipment cost for this process is therefore expected to be larger. Other large costs include engineering and construction costs, which account for a combined $9 billion. The total plant cost is estimated to be $27 billion. However, we believe this to be a conservative estimate because costs such as validation to meet cGMP standards will be accrued. The largest operating cost for the stem cell process will be the purchase of raw materials. The neuronal medium, microbeads, and cryopreservation are the most expensive components of the manufacturing process. Labor-dependent costs make up roughly 20% of the manufacturing process. This cost incorporates an estimated 100 operators who will be vital to the production. Job roles may include technician, quality assurance, quality control, microbiology, warehouse, and maintenance. For a commercial biotech facility, the estimation is again quite conservative. Facility-dependent costs will also maintain hygiene zone standards and general facility maintenance. $1.5 million is allotted for laboratory, QA, QC testing to occur for product validation and safety. Consumables will contribute a larger amount to incorporate the cost of disposable bioreactors. Waste treatment and utility costs combine for under $5,000 worth of the overall operations of the plant. The total operating cost is estimated to be $41 million. Raw materials contributes $27 million to that.
The table to the right shows the profitability analysis for the process. These values were generated using SuperPro Designer's built-in economic evaluation report. The total investment for the process is equal to $31,441,000. The return on investment is equal to 290.17% after 15 years of operation. The net profit for 1 year was equal to $91,223,000. And lastly, the gross margin was equal to about 78%. In terms of profitability, again, most of the cost estimates for raw materials and the equipment were conservative values. The selling price is also lower than the price of currently approved cell therapy treatments. The differentiation of the hMSCs would also increase the selling price as it is a more specialized product. Also, because this is a relatively new field, costs are highly compared to more established processes such as monoclonal antibody production. The unit production cost is also dependent on dose size, which is usually based on a patient's body weight. Therefore, the unit production cost is likely to be higher in value. If the dose size increases, then that would increase the cost of production, since more batches will be required. Environmental, societal, workplace, and community health safety impacts were analyzed for this process. For the environmental impacts, the component with the most waste was nitrogen gas, which was emitted at about 2,076 kilograms a batch. Excess nitrogen in the atmosphere can produce pollutants such as ammonia and ozone, which can increase poor air quality. One proposal is to perhaps recycle the nitrogen and other gases back into the process to reduce emissions. For societal impacts, it is again important to note that this process can provide a regenerative therapy for the treatment of spinal cord injuries. The process we have proposed can make about 18,915 doses per year, which directly can impact the health and well-being of patients. This manufacturing process can also provide employment to over 100 operational workers. Lastly, for workplace and community health and safety impacts, this process introduces high-risk activities and several hazards. Hazards include reactive chemical and biological agents, extreme pressures, extreme temperatures, slips, trips, falls, electrical shocks, toxic chemicals, rotating and pinching equipment. Manufacturing safety, proper training, and appropriate PPE equipment will be important to prevent and lessen the risk of workplace injury. In conclusion, we were able to create a design schema for the manufacturing of the hMSCs, from the isolation of a cell line through the growth and replication process, separation of the hMSCs from the growth chamber, differentiation into neuronal cells, and packaging the cells for shipping to a clinical site for use as a therapeutic. We were able to produce 18,915 doses per year. Some future considerations include modeling the bioreactors in SuperPro as vertical wheel bioreactors, rather than the generic ones we had chose. In addition, we could conduct more research on the costs of stem cell therapy processes to better determine the economics of the process. We can determine realistic scheduling times for the process so that multiple batches can be run in a given time. We can optimize the growth of the cells while minimizing the total amount of raw materials used in the process. And lastly, instead of assuming ideal behaviors and factors such as pressure and temperature, it'd be equally important to model this process under non-ideal conditions to create more accurate titers of the hMSCs.